The 11th annual Nathan S. Greenfield Family Lecture in Pharmacology. The lectureship, sponsored by the Greenfield family, honors the patriarch of the family, Nathan Greenfield, his wife, Corinne Sternheimer Greenfield, their only child, Dr. Rosalie Greenfield Weiss, and her husband, Dr. Raymond Weiss, and their daughter, Lynn Stewart Weiss. The family roots began in Cleveland, where Nathan, a pharmacist, operated the Wade Park Pharmacy until 1956. His wife, Corinne, was a concert pianist who studied at the New York School of Medicine, which is now known as the Juilliard. Their daughter, Rosalie, was born in Cleveland and combined the talents of her parents, first as a ballet artist, and then a pre-med student at Mather College of Western Reserve University. But her study was interrupted by World War II, where she was recruited into the FBI and served for a period of time. When she returned to Mather College, she finished a degree in psychology and eventually opened a practice with her husband, Raymond, also a psychologist. Their multi-talented daughter, Lynn, tragically died of cancer in 1971, having completed all the requirements for the Ph.D. degree in public law and government at Columbia University, except for the dissertation. We are appreciative of the family for their generous sponsorship of this lectureship, as well as a travel award each year for a graduate student who has demonstrated a special talent in presentation skills. I now ask Monica Montana to introduce our honored speaker.
Well, it's a great pleasure to introduce our speaker for the 2008 Greenfield Family Lecture, Dr. Mina Bissell. She's a world-recognized, she owns the field of the role of extracellular matrix and microenvironment in the regulation of tumor phenotype. She really has a very impressive career. She left her native country of Iran to attend college in the US, got her undergraduate degree at Radcliffe and Ph.D. in bacterial genetics at Harvard, followed by fellowships at Harvard and UC Berkeley. She then joined the Lawrence Berkeley National Laboratory, where she moved up the ranks to become the Director of Life Sciences, where she is currently a distinguished scientist. She has authored more than 300 publications, received numerous awards, including the highest honors of the U.S. Department of Energy, Department of Defense, FASAB, American Association of Cancer Research, and the Susan G. Komen Breast Cancer Foundation. She was elected to the Institute of Medicine of the National Academy of Science and has served as the president of the American Society of Cell Biology. When Dr. Bissell first proposed the idea that the tumor microenvironment is very important in shaping the behavior of cancer cell, she met a lot of resistance in the cancer communities, particular people who would prefer that we keep our focus on oncogenes and tumor suppressor. But with her pioneering work, the idea that the extracellular matrix is not merely a passive scaffold for this cell has now been widely accepted. It's also one of the areas of great interest in the cancer research community. And a couple of years ago, the NCI has made it a funding priority to look at the interaction between cancer cells and their surrounding. Despite her success, she continues to challenge convention, and that was very clear when we were talking about the role of estrogens in breast cancer. Looks like I might have a revamping of my research program based on our conversation. But that challenge of convention is going to be clear from her work that she'll be presenting. And with that, I'm going to go ahead and let Dr. Bissell.
Can you please come in front? I don't know why people don't like the front seat. Please come. There are all these seats here. You can leave in the middle of my talk if you want. I won't take offense. All right. I have a lot to tell you, and you forgive me because I found out that a lot of you have never heard me before. And so I'm going to be insulting those people who have heard me before by repeating myself. I also found out that I have a very diverse audience. I found out I have chemists and pharmacologists and biologists and MDs and whatever. And this is a big topic. So you heard, oh, don't tell me. Oh, I know why this thing is not working, because it's not connected. All right, let's see. Are you going to go? Oh, yeah, okay. So, I don't know how many of you know RAS sarcomavirus. Could somebody help me to move this? Because otherwise I'm going to be hitting it. I walk. I'm just like these Italians. If you put my hand behind my back, I can't talk. And during my talk, I move. So, all right. So this ugly tumor was discovered by Rouse in 1911. And he, as you know, is the first person who found an oncogene. He actually showed the Koch postulate. And what else he did is that he waited 50 years and then won the Nobel Prize. So I say to the students, hang in there, it may come. Now, when I came I was a chemist, as you heard, and a bacterial geneticist. I came to do postdoc in this area. And for various reasons that maybe later I have a little time to tell you, it just didn't make sense to me that we could have a single oncogene making a tumor and that we would be walking around normally. We have billions of cells, and if one single mutation could actually make a tumor, you and I would be a lump. We wouldn't be who we are. So when I had my own lab, we took RAS sarcomavirus because of some very, very old literature and injected it into embryos. And believe me, I didn't know how embryos looked. There are chairs here. And there are chairs here. I just love this, how people avoid the front row. So I'm going to tell the graduate student and postdocs, people who sit in the front row becomes leaders. So do it next time. All right, so when I had my own lab, we started injecting the virus in the chicken embryo wing, and God knows, 1,500 embryos later, my postdoc David Dahlberg and I, we both were totally nuts, and we showed that if you inject the virus into the chicken embryo, you don't get tumors. And of course, people said, well, it doesn't integrate. We showed it was integrated. They said it doesn't express oncogene. It did. Well, they said it doesn't do this, it doesn't do that. We published it in Nature, 1984. 84, yeah. And that was the heyday of oncogene excitement. And I really don't blame the field for not paying attention. It didn't make any sense in the light of what everybody was so excited about. So it took a while. And those of you who are curious can come to me and say, why is it that in the chicken you get a tumor and in the embryo you don't? I don't have time to tell you about that. But one of the things that we did do was that there are seats in front, guys. Come in. So I'm not going to say this again. It's the last time. All right. So what we did was that we took PP60 SARC. In those days, you only had LAC-Z. So we took PP60 SARC, attached it to, we didn't have GFP, so we attached it to LAC-Z. And here you see this beautiful feather, and every one of these cells could have been a tumor, but they're not. Now, if we took this embryo, dissociated it, and put it in a dish, we got mass transformation overnight. Mass transformation overnight. So what does that tell you? That tells you that in the chicken, you're getting a tumor. In the embryo, you don't. You dissociate and you put it in tissue culture, you get mass transformation. But to me, what this said is that context, i.e. the microenvironment, determines what even a potent oncogene can do. You agree? I mean, this is in base of about 10 different papers, but the question was how? Now, I was working before that on glucose metabolism, and since glucose metabolism has become such a big deal, I'm going to tell people like George Stark, who has heard me a few times, in the middle of my talk about the work I had done on glucose metabolism. So I could have taken my clothes off and given a talk to people about glucose metabolism, and they would have fallen asleep. Now, I could have put in my bathing suit and talked about chickens, and that would have done the same thing. So the question was what does one do to try and actually understand this? And having talked to the students today and having been a chemist myself, I began to think about what does it mean about who we are and what does it mean to talk about regulation. So please don't get insulted, but this is what I show to high school kids and the physicists at Lawrence Berkeley Lab. So I say when your mom and dad met, an egg and a sperm get together, and if you really want to understand why this is such an important problem, in no time at all, these cells grow and grow, and then grow and grow, and then they do this. Now, so if you really thought about it, this is tissue specificity. How did this gorgeous lady, not with lipstick notwithstanding, became who she is from a single cell? And if you really stopped and thought about it, you have billions of cells in your body. And every single cell in your body has the same genetic material, right? This, that, this. I say breast, and I immediately say prostate, so that people would listen, men, I mean. So they have a lot of similarities. So how is that possible, that a cell becomes what it is, but actually remains what it is? And of course, in the old days, people would say there is such a thing as terminal differentiation, but then somebody took a cell from a mammary gland and made a dolly, right? It came from a mammary gland. A single cell made the whole animal. So clearly, that single cell, under certain circumstances, was able to make the whole animal. So why don't you get up one morning and have your knee turn into your nose? It doesn't, thank God. But why doesn't it do it, right? So I got interested in these questions. How is tissue specificity maintained? Not how we arrived at. That's the work of developmental biologists. How is it maintained? How does one study the problem in mammals? I wish Drosophila had mammary gland. They don't. How is the program lost in cancer and aging? Because it's precisely when a cell doesn't know it's supposed to be a nose or a breast cell or a liver cell, that it piles up like a tumor and goes elsewhere when it metastasizes. So aging is the most important risk factor in cancer, right? It goes like this, logarithmically as we age. And why is that? And that has a lot of relationship to cancer, and both of those have a lot of relationship to tissue specificity. And finally, how can one use this information for therapy? So tall order. We use the mammary gland, if you will, as an experimental organism because it changes again and again in the female mammals. It goes from virgin to pregnancy to lactation to involution. It's one of the only, other than brain, that changes, but it doesn't keep changing. It's the only tissue that develops in the adult organism and then again does more than one thing. And that's shown here. So when you have the virgin animal, the cells of the mammary gland grow and invade into a fat pad. When they become pregnant, these cells grow like that. In lactating, they differentiate into these beautiful acini, or an acinus, which is one layer of epithelial cells, as I will show you, one layer of myoepithelial cells. And when you take the babies away and the gland involutes, they basically travel back here, and down here in this ancient northern blot, here you have no beta-casein, an important milk protein. Here you have more, here you have way more, and here it comes back down, right? So it's a beautifully modulatable tissue, and it's a very nice tissue to ask the question of what is the basis of tissue specificity. Now, if you look in the mammary gland itself, here is in human, you see that you have ducts, and you have in the tips of these things an acinus. So we said that the mammary gland itself is too complicated. Now, I want you to remember these branches, because at the very end of my talk, I'm going to quickly talk about a new model we have developed a couple of years ago, where we studied the invasion of the mammary gland into the fat pad. So we said we are going to understand not the entire mammary gland, but just this. It's like an organism. It's an acinus, one layer of epithelial cells, one myoepithelial cells around it, and a layer of basement membrane, and I assure you, I knew nothing about basement membrane. I learned it from my postdocs. They taught me about collagen, they taught me about basement membrane, and so that's what it is. The milk goes in here, and you say, right, these beautiful cells look like this. Very, very old pictures taken by Johann Emmerman. They are polar, they have a basement membrane, organized nuclei. Look at the way this nucleus looks. Lovely organized Golgi, secretory activity, fat droplets, and they make lots of milk. You take these cells and you say, animal is too complicated. I put them in a dish, I give them all the lactogenic hormone, give them everything they need, and I say, how do you know how to make milk? Well, you put them in a dish, and 3 days later, 3 days, the cells look like that. This is the same magnification, and I am so computer illiterate, I don't know how to turn this around. So this is back here, but look at that nucleus in relation to that nucleus, and look at the way these cells are, and they absolutely forget to make milk. You say, you silly cells, 3 days ago in the animal, you were making milk, you look like this, what happened? So to make a very long story short, we said something is in vivo that is not in culture. What could that be? Could it be the basement membrane? In the old days, we used to make this tumor in the animals. It used to be called EHS tumor. Now it's called matrigel. Why I didn't patent it, I don't know. I would have been so rich. And so you take these cells on the top of this gelatinous, laminin-rich matrix. They pull this stuff over them. In 3 days, they look like that. In 4 days, they look like this. And here is in culture, here is in vivo, and even the sizes are the same. So this question of sizeness is a wonderful problem for a graduate student. Okay, so these cells not only look good, we said, do they also make milk? So we made an antibody. In those days, we did radioactivity and stuff, and we took this stuff inside, and we took this stuff outside. We immune precipitated it. This had 30,000 count. In tissue culture plastic, you had 300. So clearly, they not only are making milk, they are not only have the right size, they are also polar. Because polarity is the most important component of your body. Everything about you is polar. Imagine that you feed the baby, the mother would feed the baby, and it gets a whole mouth of basement membrane. I mean, if the cell didn't know where to put the milk and where to put the basement membrane, you would be really in trouble. I was all worried during my trip that I was going to die of being too cold, and now I'm too hot. So I guess this, so you forgive a little bit of my striptease here. So let's see. Okay, so we now can make the human cells make these beautiful stuff. I'll show you that quickly. We can, in the, again, culture, this red stuff is milk. And these are these beautiful nuclei around 2 of these alveoli. And we have done so much in trying to understand this. But of course, I'm getting up there, and I had one of these big birthdays, and the kids did that. So we have milk, and we have a lot of milk, right? But here is laminin, here is beta-casein. This sits outside, that sits inside. How do they talk to each other? In 1980, I wrote before you guys were born, I wrote a paper in Journal of Theoretical Biology. I called this a model of dynamic reciprocity. I said, this extracellular matrix out here is actually talking to the nucleus. It's telling it what to do. It has to signal through receptors. The receptors had to talk to cytoskeleton. The cytoskeleton had to talk to the nucleus. The nucleus had to talk to chromatin. And in that was a unit. And that the unit of function in higher organism is not the cell, but the cell plus its extracellular matrix. But if you really thought about it, it's the entire organ itself. And I'll give you a little bit more example of that. So we argued that and we have shown that there are both biochemical and mechanical signaling involved. We just had a paper in EMBO journal on a single mammary epithelial cell measured by atomic force microscopy using different ECM molecules and looking at the expression of the gene. Those of you who are biomechanically oriented, please look it up. And I won't have time to talk about it, but what has happened is a couple of my new postdocs have gotten interested in this concept, and I will show you this guy's data, and I'm just going to summarize it. We have shown what receptors are involved. We have shown that a STAT5 is involved in the signaling between laminin 5 and expression of the gene. So what he shows is that if you add prolactin, which is what endocrinologists thought was sufficient. This is the amount of a STAT5 phosphorylation, and this is prolactin incubation at times. If you add prolactin to these cells, they go up with the STAT5 phosphorylation and they drop. And there is absolutely no milk. But if you add laminin, it completely reorganizes the cell and reorganizes chromatin, and when STAT5 phosphorylation goes up in a stable fashion. Stable doesn't mean constant. It's going on and off, okay? But it is constantly going on. When that happens, now you have milk. So to summarize his data, some of which appeared, so extracellular matrix and chromatin remodeling are essential for continued activation of transcription factor and organ-specific gene expression in the mammary gland. And to quickly summarize some of the other work we have done is that we postulate that tissue-specific genes don't sit on the nuclear matrix, and there are reasons for that I say that. And when we come in, so beta-casein is not expressed, you add laminin 1, which is now called laminin 111. And when you do that, now this chromatin reorganizes. What happens is CBP beta, STAT5, RNA polymerase II, BERG1, all come to the site. They reorganize, and now the transcription factors and the whole complex fires, and you have milk. Some of this came out in JBC, and hopefully the rest of it that is coming out in Journal of Cell Biology, and it's in revision, and I think it'll get in. So that's enough about mouse. So we said, are these concepts that we have developed in the mouse also relevant to human? So I had this wonderful guy who looked like this 20 years ago. This was his graduate student. They're married. They have 2 kids. And I still have the same picture, and they still look the same. So he came to me. He's an MD, Ph.D., and he came to me and he said, I like what you do. I want to collaborate with you. And I said, yeah, the good thing about you is that you're a MD, and you have access to reduction mammoplasty. So why don't we take cells from reduction mammoplasty and ask the question, is this concept of basement membrane talking to the cells is actually true in human? So he took cells from reduction mammoplasty. These things are called terminal ductal lobule unit. You take these cells, dissociate them in 2D. It looks like all the cells that have this beautiful epithelial morphology, but this doesn't look anything like that or like this. You put them in this gelatinous basement membrane in absence of serum. They reorganize and they do this. And they look, and the sizes of this are very similar to the sizes of that, as you see. So why is this? What happens actually in the mammary gland? In the mammary gland, you don't have matrigel. In the mammary gland, you don't have this gooey stuff or whatever. What is it that actually does this? So we argued that in the mammary gland, you have one layer of epithelial cells, one layer of myoepithelial cells, and then the basement membrane. And we said, what actually signals to the epithelial cells to form this? The same thing that matrigel can do too, right? So we isolated epithelial and myoepithelial cells with magnetic beads and columns and stuff, and we showed that they were pure, and we did 2-dimensional gels and protein assays in those days. And we then said, okay, if you put the cells in matrigel, they make this. So I couldn't test it in matrigel. But if you put the cells in collagen, now we can ask whether or not myoepithelial cells actually play a role in this, okay? But look at, here is collagen, here is basement membrane, and they look the same. Here, however, if you section, you see that they don't have a lumen while these cells do, right? So these cells, even though they have stopped growing, they look different. So you take the epithelial cells and you put them in matrigel. Here is sialomucin, here is epithelial specific antigen. Here is a beautiful basement membrane. Now you take these same identical cells and put them in collagen, right? Remember, they both stop growing, but look at it. They're completely inside out because the signaling is specific to the specific ECM molecules. If you give laminin, you get this. If you give if you put this in collagen, you get that. So we said, okay, in collagen they look like this. Now, could we add myoepithelial cells, which is what they see in vivo, to this mixture? What would happen? And this is what you get. So myoepithelial cells revert polarity, organize this, and for the first time, we have a double layer of the mammary gland, which is what you actually have in vivo, a beautiful basement membrane. These cells are making gobs and gobs of basement membrane molecules, but they don't know how to organize it. So we said, why is this? We isolated myoepithelial cells from tumors, and they couldn't do this. And we discovered that myoepithelial cells are the only cells in the body that make the laminin A1 chain. And that's why they act like matrigel. Now remember, matrigel has laminin. It allows the cells to do this. And laminin 1, A1 chain of laminin is required for the ability of the cells to be polar and to do all these wonderful things that mammary gland does. Now if you look in a tumor, here is in a normal, beautiful basement membrane, organized. Here are 5 different tumors. You see laminin is gone. So are these concepts, these 3D concepts, can it be used for an assay that distinguishes normal and malignant cells from each other? Now, I don't know how many of you work with breast. I know a number of you do because you came and talked to me. But how many of you work with epithelial cells, human epithelial cells? We basically don't have an assay to distinguish them, normal and malignant. It's not like chick embryo fibroblast and Ralph sarcoma virus. So we developed an assay, and the way we developed that is we took a single cells, put them in this matrigel, and here are the tumor cells, and look, those go here. These are not single cells. I apologize for this. I will tell you this is a little bit messy, but you see that every one of them look like that. Those are not single cells. They are these beautiful three-dimensional structure, and these are the tumor cells. Every one of these do that. Every one of those do that. It's like having hundreds and hundreds and hundreds of transgenic human breasts in tissue culture. Why? Because you can take them out, you can put shRNA in it, you can measure this, you could put inhibitors, you can put inducers, you can do whatever. And Mike, are you Mike or Mark? Mark. Mark knows that we have developed an assay. You heard about this? We have developed an assay on the basis of this assay that allows us to do actually genetic screens. Okay, so we said, here are the normal cells, here are the malignant cells, and we said, could we compare this to each other to try and understand them? If the normal cell comes from here, tumor cells come from there, they have been in the animal for a long time, or in human for a long time. And so we again took advantage of Ole Peterson's work in the lab. He and his professor, Per Braune, had taken cells from reduction mammoplasty, had put them in a dish without any serum, only EGF, had grown them and grown them and grown them and grown them. Somewhere around here, we took away the EGF, and we started passaging them. We call these pre-malignant because we kept injecting these into mice until one made a tumor. We put the tumor back in, is a real tumor. So all of a sudden, we have the first model of human breast cancer outside of humans without adding an exogenous oncogene. Okay? It took 20 years, believe it or not. Send it to Cancer Cell for we have also wonderful stuff. And the reviewer said, oh, it's only one model. I said, you have another 20 years to do it? Okay, so we divided it up and sent it to cancer research. So one came in 2007, one 2008. We have defined these cells, but we took these S1 cells and we took the T42 cells because they come from the same genetic background, right? Are you awake, or should I open a few doors? Go like this to your neighbor. Okay, so if you take this cell and that cell and this cell and that cell and look at them in 2D, they all look the same. You put them in 3D and they sort out. So in passage 50, already these cells have, as you see, some amplification and some deletion. These are CGH stuff done in collaboration with Joe Gray, and it's in the cancer research paper this year. Passage 110, they do this. Passage 175, but even here, when they get a little bit bigger, they do this. And all of a sudden, these S2 cells, which are a mess, are not yet malignant, but the structure is gone. And we have now an assay, if you will, for pre-malignancy. Am I going too fast? You're with me? Okay. Pre-malignancy, it's wonderful because the structure goes before malignancy sets in. And that's very exciting to us. So we said, if tissue structure is the message, we can hypothesize that if we took a malignant cell, put them in 3 dimensions, and made them remember their structure, the way those ones were in passage 50 and 75 and 100, they should think they are normal. And if we took a normal mouse and destroyed this structure, without adding an oncogene, without taking away suppressor gene, they should become malignant, right? So we did those experiments. First this. So Valerie Weaver, who now has her own mini empire at UCSF, she was a postdoc and she did flow cytophotometry, showed what was in the surface of these things, and showed that these cells had all the integrins as these, but the ratios were different. And they also had 5 times the level, for example, of EGFR. They had a lot of beta-1 integrin, EGFR. We said, right, that means that if these integrins and EGFR and stuff are high, what if we brought the level of beta-1 inhibitory antibody to the level of this? Not to 0, because they die. If you bring it to the level of this, what would happen to them? Well, we did that experiment, and that's what happens. And for those of you who have not seen this before, I say, if that doesn't boggle your mind, I don't know what does. And you see that the size is the same. They have stopped growing. Everything, cyclin D1, no matter what you would say, it becomes normalized. Clearly, they're not 100% normal because they have mutation. But if you look here, you see that these are the tumor cells. These are the normal cells. And I lost my normal movie. And the so-called normal I showed you a few minutes ago was actually this. It was tumor cells that received beta-1 inhibitory antibody. They exactly look like normal. And every one of them do it, okay? I was talking to all the DNA repair people, and with no insult intended, and they kept saying to me, it doesn't make sense. It has to be mutation. You have to have selection. I said, in 4 days, I select 10 to the 6 cells, all of which have gotten mutated. So we did the experiment. We said reversion is reversible. So you take the cells, you put them in malignant, is like this, and here is the genome. You put them with beta-1 inhibitory antibody, you get this. You take these cells, dissociate them, put them in a dish, okay? And then these same cells without antibody do that, with antibody do this. And you can do it over and over and over again. What does that tell you? That tells you that you should have actually known that from the beginning of my talk, that phenotype is dominant over genotype. They're all the same genotype, right? Look at yourself in the mirror. You should know that already. But the first time I said that to molecular biologists, I thought they'd throw me away outside. Half of them got mad, half of them clapped. So it shows you which one. All right, phenotype is dominant over genotype. So if we take these cells, these, and inject them into the mouse, no tumors. if we dissociate them with beta-1 inhibitory antibody, we reduce tumors by 70%, and the latent period is very high. So growth and malignant behavior are regulated at the level of tissue organization. And for every one of these things I told you, there is a paper, and if you don't believe us, please Google us. So the question was, the EGFR is 5 times higher in these cells. The beta-1 integrin is 6 times higher. We down-modulate beta-1, they stop growing. Why? So I asked my graduate student to measure the EGFR. We add beta-1, EGFR goes down. EGFR, so I said, what if we added EGFR antibody or EGFR inhibitors? They revert, and beta-1 goes down. So beta-1 and EGFR are talking to each other and feeding back on each other, right? There's a feedback loop between beta-1 and EGFR. Now, if you look here, these are the non-malignant, these are the malignant, beta-1 integrin, EGFR activity. You come in and you add either EGFR or beta-1, you see that all 3 get more or less like this, right? This is the relevant antibody. Now, if you look at what's here in the center, and you look here, those are the same samples in 2D. This is in 3D, this is in 2D. These are the malignant. These are the malignant with EGFR inhibitor or with beta-1 inhibitor or with both inhibitors. They stop slowing their growth, but look at that. They don't budge. What does that tell you? That tells you that signaling, again, is context dependent, that cells in tissue culture plastic don't integrate signaling the same way as they do in vivo or they do in 3-dimensional structure. And to just show you another example, for those of you who do gene therapy, here is Coxsackie adenovirus receptor. And these, you see, normal, malignant. Alpha V integrin is needed, normal and malignant. You see that they're identical. You put these in 3D, and here is normal. Look at it. The tumor remain high, and these are tumor reverted. Now, the interesting thing here is that even in the tumor, now alpha V integrin comes up in 3 dimensions. So it says both normal and malignant cells are plastic and totally dependent on the microenvironment in terms of what they do, right? So now we can do this in many, many different ways. We can inhibit beta-1 integrin, EGFR, MAP kinase, PI3 kinase, paper on AKT and RAC showing that AKT does growth and RAC does polarity. We can put in another ECM receptor called dystroglycan. We can do it with Notch, TACE, TRAP1, cyclic AMP, fibronectin, you name it. They all regulate. We can take not only just our cells, but MDA231. If we use 2 of these inhibitors, we can actually revert the phenotype and reduce malignancy. So I ask you, given the fact that these cells are addicted, if you will, to this, to this, to this, to this, to this, to this, to all of this, what does it mean to say there's oncogene addiction? Words like that drive me nuts and gets me into terrible trouble with the power structure of science, because it messes up the mind and the head of our graduate students and postdocs. What is addictive about life? These things are addicted to life. They're addicted to the whole signaling pathway. So oncogene, integrins are oncogene, EGFR is oncogene, MAP, you name it, they're all oncogenes. And they're all suppressor genes. Integrins are suppressors, whichever way you want. So form and function are related dynamically and reciprocally. And the question is, if so, what happens to metabolic pathway? If everything has to integrate, because you see, you have, again, as I keep telling you, and as you very well know, all these billions of cells. Now, if a cell in your nose apoptosed, another one grew, another one differentiated, well, you'll be a mess. You won't be who you are, right? So everything has to integrate. If this is the case, what about metabolic pathway? What about glucose metabolism? I was saying this about glucose metabolism 30 years ago, and as I told you, nobody would listen. So I just want to quickly tell you how that fits into this thing. So as you remember, the Warburg theory that is now getting famous again is that tumor cells are always more glycolytic than normal cells. So that was the first thing I did when I came from graduate school for my postdoc. Everybody was cloning and sequencing, and this thing really interested me because the literature was a mess. So because the problem was that we didn't know how to define glycolysis. So I took RAS sarcomavirus and RAS sarcomavirus transformed cells, and I showed that if you, this is the wrongest slide, but anyway, we showed that at any given time, the level of glucose transport and the glycolytic intermediates were higher in tumor cells than they were in normal, okay? And we did this by developing what I call a steady state machine, because it blew my mind the way cell biologists, including myself, do biology. I was a chemist. I came here and I was a bacteriologist. You open the incubator door, the pH changes, the temperature changes, this changes, that changes, and you're measuring everything. So I wanted to do the pattern of glucose metabolism under steady state condition. We made this thing. I still have it in my lab. There are 30 little plates with 35 millimeter dishes, 2 septums. We would come in and add 1 millicurie of C14 glucose. Those were the days that people wouldn't freak out so much. And then you would do 30 seconds to the other septum, and then you could take it out and separate it from steady state temperature and CO2, and then you could make patterns. And we immediately could see that pattern of glucose metabolism was tissue specific. Just because they use glucose, it doesn't mean they use it the same way. The same chicken embryo, the fibroblast would look like this, the liver would look like that. The mammary gland, I don't have the picture yet, would look different. So that was one lesson. The other lesson is the one I already showed you, where these were always higher in transformed cells than normal cells. And then I said, well, what else could be wrong? And then we thought, well, maybe it has to do with TCA cycle. Remember, Warburg said TCA cycle and aerobic glycolysis is wrong. But we did an experiment, and this is my old chemistry. I published it in Science in 1976. And again, you know, nobody looks at these things, but it's all right. We showed that, in fact, there was nothing wrong with TCA cycle or with any aspects of this was sort of a transfer of this molecule to that molecule, and we measured oxidative pathway. There's nothing wrong with it. If anything, is even higher in tumor cells, but there is a lot more glycolysis. Why is there a lot more glycolysis? Because, so this is the conclusion of that Science paper, that there is nothing wrong with this, but what is different is glucose transport. So the way we did these experiments is that I took these cells, brought the level of glucose transport down by taking glucose away from the medium. And it started looking like this. I put 25 micromolar glucose in here, I made them like that, and the pattern of metabolism became malignant. In a way, if you will, we were reverting the cells with changing glucose levels. And the conclusion, of course, was that pattern of glucose metabolism was dependent on glucose concentration, and that meant that the glucose level signals. But as I told you, no one would listen. So I now have this other wonderful Japanese guy who got interested in this. And what did he do? He took our human cells that I have shown you. He did arrays with one tumor cell and the normal cells. And he shows that tumor cells, the glycolytic intermediates here, are like this if you put the non-malignant cells there, right? Now, you get those reverting agents I talked to you about that are here. So when you revert, just take a look at these two bands. You see, they go here, the green, now the blue, now the light blue, now the orange. And when you put them all together, they look like this, while the tumor looks like that. Now, if you look at the TCA cycle intermediates, they're all the same. They don't change, okay? Now, he then did the experiment with 2-deoxyglucose to inhibit glucose transport or just lower glucose, and look at that. Non-malignant, malignant, low glucose. They integrate into the signaling pathway and the intelligence of the tissue architecture, which allows these pathways to come together. Are you still with me? All right, so now I show you this again, and I put glucose level as one of the other reverting agents, right? Okay. So we have used this model of formation of acids, and we have used it to show that chemotherapeutic agents behave differently if the cells are in 3D or in 2D. I was hoping to have time to show it to you. I don't think I do. We have published those in a cancer cell, this in cancer research, this one in cancer research, where we have now found new therapeutic models when the cell makes the three-dimensional structure. The genes that go down are the bad genes, and the genes that stay up are the good genes. And we went back to the patient data, and this is absolutely true. So we have a whole lot of genes that are actually doing this. We have a whole lot of genes that do the reversion, that we have actually brought them together. And this was the paper of Valerie in doing this, where they kill the cells in 2D, normal and malignant, but when you put them in 3D, the one that gets organized doesn't die. This has nothing to do with growth. It has nothing to do with the genome. It has to do with tissue structure. It has to do with hemidesmosome. And it has to do, for George's benefit, with NF-kappa B. So it goes, so when we make these things into a regular structure, NF-kappa B goes into the nucleus and prevent the cells from dying. Now, if you didn't have that mechanism in your body, you would be popping all over. Every cell in your body has the ability to die, and they have to be protected from death, okay? And the structure gives them that information. Okay. So on the basis of this, and a paper Joan Brugge had done, she came to my lab and learned how to do this, and Senthil Muthuswamy did how to do this. She had a paper in Cell, we had a paper in Cancer Cell, and Tyler Jacks and Bob Weinberg did this as a last sentence in their review. They did a lovely review. I was delighted. They said, all of a sudden, studying cancer cells in 2 dimensions appears to be quaint, if not archaic. Now, those of you who know Bob Weinberg, you know that's his language. He writes magnificently. So I was at Whitehead Institute, and I said, Bob, I'm delighted you guys came around, but I have a correction. It's not all of a sudden. It took 30 years. Now we can use the assay in looking at MCF7. We have developed a new assay on top of the gel 5 years ago. The cells, MCF7 do this. MDA231 cells do this. So we said, hey, could we use this as distinguishing tumor types, breast tumor types? So we did 25 of these cell lines that Joe Gray had used 2D, and he had done arrays, and we are now doing the rest of them. They fall into 4 categories. We call them round, mass, grape-like, and stellate. So here are the round. Here are the mass. This is 2D, this is 3D, and this is the staining. Here are grape, and many of these are metastatic. And here are stellate. Now, we did arrays in these cells, okay? And now, lo and behold, the morphology tracks with this. So if you don't have money, you don't have to do arrays. Just look under the microscope on the top of a gel, and your cells will sort, okay? So conclusions are that 3D models of cancer cells are robust surrogates for breast cancer. Now, do basement membrane and tissue architecture regulate form and function also in vivo? I don't have time to tell you this. It's another 20 years of work, wonderful collaboration with Zeno Werb. We started working together, and we first started in my lab looking at involution. We said, if this 3-dimensional structure required, you see, this thing. We said, if this thing required the basement membrane, as I have shown you, when the function gets lost, it means that ECM and basement membrane have to go. So we looked here. We showed that if MMPs, metalloproteases, don't come up, mouse doesn't involute. If we have tissue inhibitor of metalloproteases to an involuting mouse in one side and put the regular on the other side, the side that has TIMP doesn't involute, okay? So in order for the milk to get lost, you have to mess up the structure. And then we made a mouse, a transgenic mouse, with the MMP, and we show that that function gets lost. Even in the heterozygotes, the basement membrane is gone. And what happens to these mice? Remember, we didn't give them an oncogene, right? We just messed up the structure. They get tumors. The tumors are genomically unstable. This came out in Cell a long time ago. And then the question was, do the cells get tumor because MMP selects an already mutated, initiated cancer cell, or do MMPs by themselves cause genomic instability, right? So this guy, whom I still miss, he's at Mayo Clinic. He has a nice paper in Nature, took many years, as you can imagine. We developed a model in culture to do a rapid analysis of this in culture so that we won't have to do it in the mouse. We did a TET regulatable MMP3, infected the cells, and then added TET or removed TET, and MMP came up, and we said, does genomic instability sets in? Yes, look at that. So this is this important assay that Dr. Stark knows very well about. In fact, I called him at the time when I was doing the PALA assay. And so here are the non-malignant, here are the malignant. The genomic instability goes way up. This is the CAD gene. Here is fish, showing that they get all amplified. And they not only do that, they also do EMT. And we showed that this is because of a splice variant of RAC. When you make MMP and the MMP messes up the cells, RAC gets spliced to RAC1B. RAC1B goes up when you turn on MMP. When you wash, it goes back down. We showed that it had to do with reactive oxygen species. I'm sorry I'm going so fast. If you're interested, you have to read the paper. You can measure the mitochondrial ROS by this assay and by a lot of other assays. We showed that it was the mitochondrial ROS. And again, to make a very long story short, we show MMP3 messes up E-cadherin. We published that in '97, actually. The E-cadherin either first makes the RAC1B or changes cell shape. We have a really cute paper showing that the cell shape change is absolutely required for EMT. And then if you make the RAC1B, now you make mitochondrial ROS, you do EMT, and you do genomic instability. And whether or not these two things are related, we don't know. So organ specificity is plastic, dynamic, and context dependent. Everything about you is in motion. Everything about you is balance. If something goes down and then you bring the opposite up, you'll be okay. It's not the absolute level of something, but the balance of these things. And so it is not absolute. So now, in the last 7 minutes, I'm going to talk about whether or not, now that we have spent all this energy telling you about the asanas and what we learned about the asanas, what kills patients, of course, is metastases. We learned so much about cancer and new therapeutic agents for cancers, or a whole lot of other information about the pathways just studying asanas. So the question was, here is a whole mount of a mammary gland of the mouse in the virgin. Here is higher magnification, and this one you had already seen. So when the mammary gland starts, it starts with a little onlaga. This onlaga, when the hormone comes, it starts invading into the fat pad, but it never breaks the boundary of the fat. And we said if we could understand how this thing actually invades, we could understand how the tumor cells hijack it, the same way that we talked about the other stuff, right? Okay. So we wanted to really have an assay where we could measure this. I have about 15 papers on branching morphogenesis, and when you put them in either collagen or basement membrane, the cells go every which way. It takes 10 days before you see if they branch or not. So how do you do all these things? How do you do it in a statistically significant way? How do you do it quickly? How do you do all of those things? So this wonderful lady, who is now at Stanford, at Princeton, excuse me. She's assistant professor. She's a bioengineer. And this lovely gal who was my technician and now is my graduate student, both brilliant women, And they decided that what they're going to do is they're going to do a micropattern. They took advantage of a paper I had published in '97 again, where you put one layer of collagen, we put either MDCK cells or mammary cells on top, and then we put another layer of collagen, and these things made a lumen. So when they do that and they make this micropattern, they make one layer of collagen, they put the cells in these, and put another layer of collagen, so every one of these have a little lumen in them, right? So they now binarize and stack 50 of these. If you just have one, you see, it just does this, another one does this. You put 50 of these on top of each other, and they do this. And you say, why is this? The cells are everywhere. Why do they come out just from the tip? And is it because of EGFR? No, it's here, here, here. Is it because of growth? No, BRDU is here and here. Is it because of MMP3? No, it's here and here. So what is it? So she started playing around with these, and we made a square. They come from the corners. We made a circle. They come from everywhere. And the one I really like is the Y here, Y. So here and here and here. So I took her with me to these wonderful people, a postdoc of Dan Fletcher, who are theoretical biologists, bioengineers, etc. And I said, model this for us. We have this. What on earth is going on? And they said, you have a diffusible inhibitor. And I always thought I had to have an inducer. They said, you have to have an inducible inhibitor. And I said, OK, if this is the case, we have to be able to test it. Because if the diffusible inhibitor is here, and normally you get branching here and here, if we put two of them like that, it should just do here, 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 and here, and here, right? So we tested that, and that was the case. And it was one of the nicest, most satisfying experiments in my life, because we did the experiment, they modeled it, we tested it, it was right. We were about to open a bottle of champagne when we said to ourselves, what is the inhibitor? It lasted exactly 30 minutes. So you go on. So there I put my old hat on, and remembered that Charles Daniel, a long time ago, had done an experiment in the mammary gland where he put a pellet of TGF-beta, and I said, could the inhibitor be a TGF-beta? Now, it's unlike what you guys know in tumors, right? In tumors, TGF-beta makes them go faster. And I was saying this morning to people I was having breakfast with that I did the first paper on that in 1985, where we showed that TGF-beta does both. OK, so we did TGF-beta, dominant active, dominant negative. You don't need to know. Whatever. It really tracks. And those of you who are interested, this came out in Science already 2 years ago. But one of the things that had always interested me, people talk a lot about EMT, and Bob Weinberg has gotten very interested in this. It's all over the place. Everybody talks about EMT. It never made any sense to me. I said, unless it happens in vivo. If it's going to do anything good, it probably has to happen in vivo. And I said, these are branching, these are invading. Could there be a transient EMT? And so we took a little vimentin promoter, attached it to GFP, and look at that. So the thing that correlates with where these things come out, there is a little transient EMT. And I know a lot more about this, but my postdoc swore me to secrecy, so I'm not going to tell you what else. I'll come back in 2 years and tell you. OK, I want to finish with this picture. So the top is where water came into the shore, taken by NASA satellite. The bottom is coral. Now, do they look like that whole mount mammary gland I showed you? Yes, yes? Now the question is, why does nature do this? It does the same form over and over and over again. So I like to submit to you that we know a lot about the sequence of the DNA. We know a lot about the alphabet of the DNA. We know nothing about the sequence and alphabet and the language of form. We know nothing about language of form. The reason that your nose looks like your nose There is a wisdom in it. It's intelligent design of the developmental biology, all right? I don't believe I'm not on the other side of evolution, okay? There is something very important about the evolutionary way you all started. Your nose, your mouth, your eyes, there is wisdom in the architecture of your tissues. And what I like to say is that this is an opportunity for the young and the passionate old, that's me, a whole new horizon to explore, and you should go to it. I suggest to you that there is zero reason to be arrogant in science. Arrogance kills. And we have so many famous people who are so arrogant. What is the reason for that? That's why people like George are so wonderful, and that's why Judith Hochman was so wonderful. What is the reason? We know so little. We know a lot, but we know very little. Everything I have told you about mammary gland is going to be true about lung, it's going to be true about colon, is going to be, and everything is tissue specific. Remember, you have BRCA1, you get breast and ovarian cancer, you don't get a nose cancer. You have APC, you get colon cancer, and yet you have it in every cell of your body. Cancer is a tissue-specific disease, and I argue that you can have mutation until you're blue in the face, unless the structure goes. And that's why cancer takes 40 years. You have mutation, 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 mutation. Nothing happens until the structure goes. All right, so I love this cartoon. I say to people, that used to be me, and that was the authorities. Unfortunately, I have become an authority, so don't listen to me either. Just question your professors, question yourself, and question your textbooks. They can kill. Half of them are really not true, and half of them are totally selected. Okay. This is Lawrence Berkeley Lab, where I did my work. It's not Lawrence Livermore, guys, okay? You see that beautiful Golden Gate Bridge down there? This was the dome of the first atomic, where they accelerated the first atomic bomb. They kept the dome. We don't do defense-related stuff anymore, but Livermore does, okay? So blame them for anything. Don't blame us for it. So we have this on the top of a synchrotron radiation, so if you do one of the world's brightest source of synchrotron radiation, so if you guys come to LBNL, you can do a structural biology. And this being 4 o'clock, 5 o'clock, this is still too early for me though. What does this look like, guys? Mammary gland? You'll never forget Lawrence Berkeley Lab again, okay? There's the rest of my group. I think I mentioned many of them. And there's my website. And I tell you, I thank the Office of Science and the Office of Biological and Environmental Research for funding. I couldn't get money for 15 years. You think you're having a hard time? Hang in there. You have to go find yourself something else in order to get money to do your passion. Stay with it. You only have one life. Stay with it. Do something good. So now they give me too much money. I could have used it 20 years ago. Now I have 25 kids running around my lab doing whatever. And I said, thank goodness for multiple sources of fund, because, you know, NIH or any big conglomerate, like the oil companies, they can clone themselves, right? Now I'd like to finish again with this. This beautiful woman was modeling this a week after her breast was removed. She died, and it was too late for her. And I have a lot of passion about this, because this is my beautiful granddaughter. When she was 5, she's now 8. I don't want this for Chloe. We have still a lot to do. Thank you very much.
So we have time for 1 or 2 questions.
She's prettier than the rest. I am happy to answer questions. I used to tell people that I will give them a hug. I'm getting too old, so people are not volunteering anymore. Okay, questions. Did I come? Oh, yeah. Yes, honey. So did I give you some good reason to think about what to do? Remember you asked me that question at lunch?
Yes.
Those of you who didn't come to lunch, you lost that.
There's lots to think about. Thank you very much for the lecture. I was just wondering, do you, in your mind, do you see any way for information then that we've been getting from 2D to, is there any way for them to meet?
Of course.
From 3D?
Of course.
They're coming up with the same conclusions about selectivity.
Of course, in
and
some cases.
regulation
Of course.
and their outcomes.
But where would I be without PI3 kinase having been cloned and without the STAT5 being understood and whatever? You use those molecules. I have used whatever is in the literature, all right? And in some instances, you see that the two come together, but it's not in every case. So in order for you to be able to use it, and I say this to everybody, now remember our Nature paper is all on 2D. Okay? Why is it in 2D? Because we wanted to develop a wound environment so that this would go faster. I didn't want to make the mouse pregnant and then have involution and then have it again and have it again and have it again. Every 2 years for one assay, you can't do that, right? So because we had shown it in vivo, because we had shown it in three-dimensional structure, we then said, okay, we could do it in 2D. And when we did it in 2D, we got EMT and we got genomic instability, and that, in fact, could explain what was happening in vivo. Of course, but you have to do it thoughtfully. It can't be done because it's there. And what you learn from mammary gland, you could extend it to brain. You could extend it to lung. Every single cell in your brain touches a proteoglycan. People who work on sugars are going to inherit the world, I tell you. It's going to be so complicated. But the details are going to be different. For years, it has driven me nuts when people stand up there and say, A goes to B, goes to C, goes to D. It's true that you have downstream, but you also have upstream, okay? In some cases, you have feedback inhibition, and in other cases, you don't. So what I say in the mammary gland is true for skin, is true for ovary. But say E-cadherin goes down in breast cancer immediately. E-cadherin goes up in ovarian cancer immediately. And then it's very late that it goes down. So you have to develop these systems for every tissue and for every condition. And then you have to put genomic susceptibility on top of that. Everybody says, oh, my grandmother smoked until she was 100 and she didn't die, so I can do it too. Well, it doesn't work like that. Your grandmother probably had something that, So there is differences between people, and there is clearly differences between tissues, right? So, yes, sir.
I have one quick question. When you show at the beginning all the pictures about how the basal lamina could make changes in the tumor tissue and becoming more like normal cells.
Yes,
just by
it wasn't basement membrane. It was signaling of the basement membrane. Because remember, those tumor cells were inside a laminin-rich matrix. That wasn't sufficient. In fact, if anything, the basement membrane and the gel is like a blotter. It brings up the differences between normal and malignant. So in this case, we were changing signaling of the basement membrane.
Then
Yes?
my question is more, can you envision that, can we do treatment to,
I'm glad you asked. Okay.
in this model of breast cancer, do a mechanical treatment in which by any modulation with plastic and the signal that you have in the basal lamina, you will reorganize the tissue in the breast and try to make them be more normal and then advance the cure.
So a lot of people say to me, what good does it do to revert malignant phenotype if one could revert it, which I'm not sure necessarily we could. So I will answer your question in two ways. One is that we use the beta-1 inhibitory antibody in the animals. And the way we did that is that we took many different, say 6 different breast tumor cells, put them on the top of the gel, and allowed the tumor cells to form this and the normal cells to form that, okay? Then we treated them with beta-1 inhibitory antibody. Then we showed that the normal don't die and the tumor die, okay? Then we took these tumor cells and put them in the animal and allowed them to go on for a few days. So they are more like what they were. And then we came in and injected beta-1. This came out last year in Cancer Research. And what we find is that under these conditions, both in culture and in vivo, we don't necessarily revert, but we kill the cells. And we actually can treat them that way, all right? But the idea of whether or not reversion theory or differentiation theory actually works is been shown in some cases in lymphomas, right? You give them retinoic acid, you give them interferon, and these mutated cells of things goes in remission. And the way it goes in remission is by becoming normalized and differentiated. And after 10 years or 20 years, it comes up again, and then you add more or you add something different. And believe it or not, they do it through beta-1, even though these are signaling molecules. It's quite interesting. So I would say that in the long run, the concept is important. And what is most important is that we now should go and use therapy for the microenvironment. Because we know very well, like in esophageal cancer or the Barrett's esophageal cancer, you have a field where everything is messed up. So you take this tumor, another tumor pops up. You take that tumor, another tumor pops up. Is because of the years of wounding, of the years of whatever, that it starts making the microenvironment to be actually able to cause tumors. In our transgenic animal, the thing that happens first is that the stroma starts going. It actually upregulates the endogenous MMP. It has a lot more collagen. It makes a reactive stroma, and then eventually you get tumor. And Hal Moses has done a lot of lovely things. They have a model where they mutated TGF-beta receptor in the fibroblast. Do you know that work? It was in Science, and I wrote actually an editorial for it. And then what they show is that the mutation in the fibroblast causes tumors in the epithelium in 2 tissues only, which is also, again, tissue specific, okay? More and more is coming out that cancer is the problem of the organ. I wrote a review in 2001 in the first issue of Nature Reviews of Cancer, and I argued that tumor is a new organ. It constantly, however, remakes itself, depending on what it hits in the microenvironment. So you have to treat the tumor, but you also have to treat the microenvironment. You guys are getting tired. There is a reception. I'll answer one more question and we go. Somebody else had their hand up?
I had the same question.
All right.
You know, I have
Yeah.
something related, I guess.
Yeah.
More clinical one. So you were showing work with beta-1 integrins,
Yeah.
metalloproteinases, but inhibitors of this, molecules,
Yeah.
we know they don't do that well in clinical trials.
Well-
I think not enough.
Well, no, well, we can talk about that, all right? Now, one paper that we just published this year, again with the same colleague who was doing that, who's a radiation oncologist, is that we showed again in the 3-dimensional culture that we could combine these inhibitors with each other, and that we could combine it with radiation. So in a situation where we make a tumor, and we give 8 gray of radiation to the mouse, okay? In order to get inhibition of the tumor, we showed that if you use beta-1 inhibitory antibody, we could bring the dose of radiation from 8 to 2. Imagine what that could do for women who get radiation therapy if we could learn how to do that. So we are now trying to humanize the beta-1 and to try and take it to the...
Beta-1 general or alpha-6 beta-1 or alpha-2 beta-1? Which beta-1?
You know, Genentech is working on this, and as are a lot of other people, and you need to take it in your company, and you need to do it. I'm getting old. All right. Okay, guys, thanks a lot.